Today, I'm going to talk about the suitability of phages for phage therapy. But let me start with a disclaimer. This will be a lot of science, I warn you. And to make this palatable for a very broad audience, I have to generalize. I will not be able to address the exception to the rule. So if you have a feeling that I skip over a topic that is very important to you, ask it in the chat or get in touch after my talk. And second, my drawings are intended to illustrate scientific features, and they're not drawn to scale. With that out of the way, let me start with a view to history. Phage therapy was a very hot topic 100 years ago, after Felix Derel successfully treated patients with Shigella dysentery. Many scientists, including many medical doctors, started to work with bacteriophages and tried to cure patients. And their results were a really mixed bag of beans. Some reported spectacular recoveries, and others reported almost uniform failure. With hindsight, we really know the challenges these scientists faced. Basic concept of science, like that DNA is the hereditary material and not protein, were simply not available to them. And we really have to admire the diligence and the persistence they did in investigating the topic. Now, 100 years later, we can really step on the shoulders of these scientists, and we think it's time that phage therapy becomes a hot topic again. In the last session of the Academy, I showed you this basic lytic growth cycle, and I told you that the destruction capacity of a bacteriophage in this lytic cycle can be harnessed for phage therapy. Today, I would like to go into a little bit more detail what such a lytic growth cycle is about and show you how we want to use it in phage therapy. The first step is the adsorption to the bacterial cell, and this provides a lot of specificity. Not only specificity between different bacterial species, but also within a bacterial species. A receptor gene in the genome of a bacterium doesn't necessarily need to be on the surface of the bacterium at all the time. Once we tackle adsorption, the phage genome has to be injected into the protein, into the bacterial cell. And the obstacle is the bacterial cell wall. And you can imagine that you need a different strategy to go through this cell wall than to this cell wall. So that generates another specificity point. Once you're inside the cell, there is immediate danger because DNA is food. And every bacterium has enzymes that degrade DNA from the ends. And the bacteriophage has to protect its genome from this degradation. Now, there are several ways a bacteriophage can do that, but the most elegant one is to simply recircularize the genome, and then you have no free ends, and the exonucleases don't do any harm. Also, instantly after the injection, the host machinery starts to transcribe and translate the genome. And this is also specificity. The bacteriophage genomic information has to match the ribosome of the host. Otherwise, this protein expression will not take place. So we produce the first phage proteins in this step. In the next step, the genome of the phage has to be amplified. And that is first very simple. Out of one genomic circle, you replicate 2, out of 2, 4. And then the phage switches the strategy to so-called rolling circle mechanism that generates very, very long DNA molecules where multiple genomes, 10 or more, are in one DNA molecule. In total, about 200 copies of this phage genome are produced. And from these copies, these 200 copies, you can now really start a lot of gene expression. And all the late functions that code for the head, tail, the tail fibers are now produced in massive quantities. And then the phage needs to package the DNA into the head. And for that, it has a protein called primase that grabs onto this long DNA molecule and starts to pump them into empty heads. And either when the genome is full or when the head is full, this protein makes a cut and hops to the next empty head. And this is really a conveyor belt. This happens very rapidly and very processively. Once the heads are filled, then you can assemble the entire phage particles. And that really needs to be in sequence. It doesn't make any sense to attach the tail to the head if there is no DNA in the head. That would lead to waste. And finally, as the bacteriophage accumulate within the cell, the bacteriophage produces a cell wall degrading protein, and that needs to be transported through the inner membrane to reach the cell wall, where it will start to degrade the cell wall. And eventually, by internal pressure, the bacterium will burst open and release the progeny. Now in nature, we find bacteriophages who exclusively have this lytic growth cycle as amplification strategy. And we aptly call them lytic phages. And how can we now harness this destruction capacity in phage therapy? I would like to give an example. Assume we have a bacterial culture. If this is pathogenic and somewhere in the body, you really have an infection. So we now add a high dose of a lytic phage and let the phages to work. And the optimal outcome of such a treatment would be that we get a 2-log reduction. In nature, you never reach 100%. What that means, we have a bacterial culture that is still 10 to the 6 CFU per milliliter. And if that doesn't have the pharmaceutical effect or the treatment effect we expect, then we simply add a second dose of the same lytic phage, and that will lead to another 2-log reduction. And if this 10 to the 4 is still not sufficient, then we simply add a 3rd dose or a 4th dose or a 5th dose. Eventually, we will manage to reduce this bacterial population of pathogenic bacteria, either alone or with the help of the immune system of the patient. So that's how we plan to treat. Now, in nature, we do not only find lytic bacteriophages who can only do this amplification cycle. There are also bacteriophages who have another way to amplify their genome, another way to grow. And the decision they have comes here at this stage where early protein synthesis already took place. And the phage now has a choice. Either I go this way and use this bacterium to make about 100 copies of myself, or I enter a symbiosis with this bacterial cell, integrate myself into the genome, of the bacterial cell and allow the bacterial cells to amplify me. And I can make, out of one copy, I can make billions of copies of myself. So that sounds like a very, very clever strategy. Every now and then, if situation changes, such a situation can also be used. And then the phage excises its genome again from the host and ends up here, and then can go back and forth. Both ways are option. Now I have to make a little bit of nomenclature because that is historically grown and is not very intuitive. A phage who has those two options is called a temperate phage. The genome of this phage, once it is integrated into the host chromosome, is called a prophage. And the bacterial strain that carries a prophage is called a lysogen. And the process that leads to this integration is called lysogenic conversion. And everything together is called lysogeny. I'm going to use these terms during my talk, and I hope I don't confuse you too much. But let's go back here to this step where this decision takes place. This is a genetic switch that has been studied very intensively. It's really surprising that a handful of phage proteins can determine such dramatic decisions to go lytic or to go lysogenic. The decision depends on the protein synthesis rate and the protease activity, so protein sterility. And it makes a huge difference if the bacterial cell is growing exponentially, or if it's starving, or if it's in stationary phase. But in general, under optimal condition, an infection results in about 80% lytic growth, and 20% of the phages decide to grow lysogenic. Now let's look at that in a little bit more detail. If you want to enter into a symbiosis with the host, you have to make sure that you do not do any damage to this host. And that is the guiding principle. Now, all the functions that lead to lytic growth, like the amplification of the genome and the expression of all the late gene, and especially the expression of the enzyme that degrades the cell wall, that has to be stopped. And this is done by repression. So the phage genome encodes a repressor that then is synthesized and binds to all the regulatory sequence directly or indirectly to switch all these lytic functions. Now, once that succeeds, and that's not easy, but we assume it does, then you have an immediate problem, and that problem is cell growth. If this is all, then the phage genome would be rapidly lost out of a population. So you have to do something to make the phage genome stable. And that is done with an integration. Another phage enzyme will pair the two circles and then recombine them. And if you recombine two circles, you get a co-integrated big circle. And that is also a place for specificity. The phage and the host have to know each other, otherwise this integration will not work efficiently. Once you have done that and you're sitting in the chromosome, this is called a prophage. You have, again, the problem with cell division. Now, it is solved for the DNA that is sitting in the genome and is replicated passively, but it is not solved for the repressor molecules. If it would happen like here, the repressor only ends up in one cell, then there will be problem in this cell. So one solution possibly is that you simply fill the entire cell with repressor molecules. And then each daughter cell inherits so many repressor molecules that the whole thing is stable. But that is a really, really bad idea that takes way too much energy, and you shouldn't do that to your host. So the phage has to find an alternative mechanism. And that is a really very elegant autoregulatory control system. So the repressor molecules will start to accumulate in the cell. But once a certain low but significant concentration is reached, the repressor will bind to the DNA to its own regulatory sequence and switch itself off. So this is the maximal concentration of repressor that is reached in the cell. And if now the bacterium divides, this repressor concentration will sink. And then what will happen is that the first repressor molecule that falls off the DNA is the one that controls its own repression. We will have synthesis again. So the repressor concentration increases to this specific level, and then it binds again. And that's how the phage manages to stably maintain lysogeny, to suppress at all time the lytic functions, but do not do any damage to the host. And so all bacteria that are resulting from this have this system in there. They have a constant, slightly fluctuating repressor concentration that maintains lysogeny. Now, every now and then, it happens that one cell no longer has a repressor because a mutation occurred, DNA damage, or the cell is starving, the protease activity changed, so the repressor is degraded and no repressor is synthesized. There are several ways. But if this occurs, this immediately induces the prophage. What will happen that early gene expression starts the same way as the genome would just have been inserted. And then the integration process is simply reversed. Enzymes cut out the prophage from the phage host, and that is simple enough. From the big one, you suddenly just make two small circles again. And then this circle, we are coming back to exactly this situation here. The phage now has two options, but as the option of lysogeny is no longer possible because the repressor cannot be synthesized, the phage will go into a lytic cycle and produce progeny particles. So when I draw a lysogenic culture, there will always be free phage particles in the environment. And now this system altogether becomes really, really interesting and really very stable. For example, what happens if such a free phage particle adsorbs to a cell where its own prophage is already sitting inside? Then the genome of this phage will be injected into the cell, but instantly repressor molecule will recognize it, will bind it, and will switch it off. And this genome eventually will be lost. It will never be expressed. And that we call immunity. A temperate phage cannot grow on its lysogen. It's simply immune to superinfection. Or what will happen if a bacterial cell manages to get rid of the prophage? We call that a cured cell. That could be a problem because such a cured cell could grow and take over the population, but a free bacteriophage will recognize it, inject its genome, and then either go through a lytic cycle producing more phage particles, or it will re-lysogenize this cell, so convert it back into a lysogen. So in a lysogenic situation, we do not observe cured cell. Or let's assume we have an invading bacterium similar to the one lysogen is. Then the bacteriophages, the free bacteriophages, will find these invading bacteria and either convert them into free phage particles or convert them into a lysogen. So they're simply assimilated. And now I have a final point about lysogeny. This is a true symbiosis. It's not that the phage is simply a parasite and allows the host to amplify it, but temperate phages bring new genes to the host. And the lytic genes, they constantly have to be switched off, but other genes, they can freely be expressed also during lysogeny. And so we find phages that bring metabolic genes or replication genes, translation genes. We find some that make actually phage defense, so they defend against other infecting phage particles. We find phages that carry antibiotic resistance genes, and we find phages that contain toxins. And some of the nastiest toxins we find in nature, like Shiga toxin, are actually sitting on temperate phages and can be spread via lysogenic conversion. And if you now have already a little bit of bad feeling, let's see if we can use a temperate phage for phage therapy. So we repeat this experiment we already did with the lytic phage. We have a bacterial culture with about 10 to the 8 bacteria. If they're pathogenic, they really make an infection. And we add 10 to the 9 temperate phages and allow them to work. What we will observe is that we get about 80% reduction. That's the phage particles that go through a lytic cycle. But that will leave us with 2.7 CFU per milliliter. And the bacteria that are surviving this treatment, there are now lysogens. And if we attempt to add a second dose, then this lysogen is simply immune to the action of the phages we add, and it will regrow, and the infection will persist. So this autoregulatory control rule is very, very efficient. It provides immunity. Self-infection doesn't take place. And that is really the reason why we say temperate phages are not suitable for phage therapy, because they do not work. But you will hear all these arguments that we do not use them because they carry toxin genes or they carry antibiotic resistance genes or they spread them via lysogenic conversion. That are really only secondary arguments, because also a temperate phage that doesn't have a toxin gene or doesn't have an antibiotic resistant genes does not work in therapy. So now we've seen lytic phages do work, temperated phages do not work. Can we actually distinguish them? And if we look in the electron microscope, we find that doesn't work. We have seasonal virus morphology, temperate and lytic look the same. Podovirus morphology, temperate or lytic look the same. Myovirus morphology, temperate or lytic look the same. So in the electron microscope, we cannot distinguish them. If we look at plaques, it's a little bit better. We find the lytic phage makes a clear plaque, while the temperate phage makes a donut-shaped plaque. In the center, the lysogen starts to grow. That gives the plaque disappearance. Unfortunately, not all temperate phages make such nice plaques where the donut form is visible. So plaque gazing works sometimes, but not always. What we really have to do is bioinformatics. we have to sequence the phages and then analyze them with bioinformatics tools. And we can look for control regions, we can look for repressors, we can look for integrases, we can look for control units and also antibiotic resistance genes or toxins and so on. And with this, we can really distinguish lytic phages from temperate ones. And now a question, of course, is, okay, temperate phages can't be used. Can we somehow convert them into a lytic phage, like just chop off this lysogenic growth thing? And with our modern gene technology, we actually can do that, but it's really not easy. You have to get rid of a lot, like the repressor and all the regions it binds to, the autoregulator control loop. We have to remove the integrase and all the sequences associated with this integration. You have to look for anything potentially dangerous. And while you delete all these things, you have to make sure that the control regions of the lytic phage are actually still intact. Otherwise, the lytic cycle would not do. And as these control regions are overlapping, this is not very simple. You really need to know what you're doing. So if I have some time, yes, I guess I'm doing fine. I want to come to a completely different topic, transduction. And I don't know how many people have heard of transduction. It's a form of horizontal gene transfer that happens in prokaryotes. And the definition is that bacterial DNA is transferred from one bacterium to the next via a phage particle. And transduction has a really, really bad reputation. And I would like to try to correct that a bit. So answer the question, is it really relevant for phage therapy? To do that, let me show you an experiment. And I know it's a bit complicated, but bear with me. I take a lytic phage, grow it on a host that has no antibiotic resistance gene, then purify the phage, make sure any bacterial DNA is removed, and use that phage to infect another host that has no antibiotic resistance gene. And then I plate this whole mixture on a plate that contains an antibiotic and ask, do I get antibiotic resistance bacteria? If I do this experiment, the answer is no. I get 0 antibiotic resistant colonies. And that can be quite easily explained. Spontaneously, this bacterium doesn't become resistant to the antibiotic, so the antibiotic cannot come from this bacterium. The lytic phage doesn't have an antibiotic resistance gene, and the donor doesn't have. So where in here is an antibiotic resistance? So nothing can happen. But when I tell you this was a very highly successful transduction experiment with about 10,000 successful transduction events, I hope you're a bit puzzled. Now we just change one thing in this experiment. We switch from a lytic phage to a temperate one. We do all the things the same and ask, are there any antibiotic resistant colony coming out here? And what we find, oh, yes, massive, 10 to the 5 per milliliter. So where are they coming from? And the only explanation is they come from the temperate phage. The antibiotic resistance gene already sits on the genome of this phage, and by lysogenic conversion, the bacterium becomes antibiotic resistant. But this is lysogenic conversion. It is not transduction. And now we do the transduction experiment like it is described in many textbooks. We simply change one little thing, namely the donor is antibiotic resistance. We do the whole thing again, and we analyze, are there any antibiotic resistant genes here? And the answer is yes, we find one. And so we can calculate a transduction frequency. An antibiotic resistant gene in this experiment is transduced with 10 to the minus 6 per gene per generation. So a very low frequency that in the phage particle from the donor, an antibiotic resistant gene is coming to the new host. How is this mechanistically happening? And for that, we have to go to the lytic growth cycle and do the packaging. I told you before, a primase, a phage enzyme, pumps DNA into empty heads. It grabs onto the long DNA molecules that have been replicated. And the question, what happens if this primase runs out of DNA? This would be really bad for the phage, because then this entire conveyor belt, this entire packaging machinery, which very processively at very high speeds is running, would completely break down. And that would interfere with the efficiency of phage replication, amplification. And so this enzyme simply said, I have to go on, and it latches to the next free DNA end. And this actually makes sense, because the next free DNA is probably another phage molecule that can be packaged. So like this, the packaging continues. But what I haven't told you until now, in this cell, we also have other DNA fragments. The genome of the bacterium at this stage of the replication cycle has been fragmented. And the genome is about 50 times larger than the bacteriophage genome, or the genome of the bacterium. And these fragments can be really huge. And so what can happen is that anytime by mistake, the primase simply latches onto a piece of bacterial genome, and then starts to package, where the first particle will have something mixed, but any processively packaged phage particle will now suddenly have bacterial genome in its head. And you fill them up, and then you complete them into phage particles, and such a particle is called a transducing particle. Now, this happens with a relatively low frequency, and it happens completely random. The fragmentation of the bacterial genome is completely random. The length of these fragments is completely random. And that means any specific bacterial gene is packaged into the phage head with approximately the same random proportion. What happens if such a transducing particle finds a new host? Well, then its DNA will be injected and end up in the cell. The nucleases will start to grow, start to work. DNA is food, but any bacterium has a recombination system that pairs matching DNA. And as this piece of DNA comes from a sister bacterium that is closely related, the homology between them is given. And so you can have homologous exchange. And in the end, you have a bacterium that looks exactly like the one we had before. So what's the point of this? Well, first, I want to ask the question, why is this transaction frequency so low? Why do you only transfer 1 in a million? And One answer is the packaging mistakes are rare. So only about 1 in 100 bacteriophages is actually a transducing particles. Then the packaging capacity of the phage is very limited because the host genome is 50 times larger. So only a small, only few genes can be packaged at the same time. Then if you have 1 transducing particles and 100 active phage particles, then if a bacterium is infected by 1 active and 1 transducing particles, you will never see it. The injected DNA can be degraded, and the recombination has a probability of only 50%, and that really limits this efficiency. But transduction is a very, very important process and is really underrepresented in our view, because it is a bacterial repair system that really functions on a global scale. And I would like to explain this a bit with this slide. If you have a bacterium that has a mutation that blocks it from utilizing glucose, then all the progenies of this bacterium will carry the same mutation. And if glucose is the only carbon source, these bacteria will be starving. Now, if you have an invading bacterium that is a wild type, can happily grow on glucose, this bacterium will grow and very rapidly outgrow this starving bacterial population. So there's a danger that they simply will be lost. And now we bring in a phage. A phage grows on this wild type bacterium, produces a transducing particle. That transducing particle absorbs to one of the mutant bacteria, injects the genome in there, and it will be integrated or not, so with a very low probability. But then this bacterium that receives the transducing particle can fix its mutation. That is not directed, that is completely random, happens only at the low frequency. But in evolutionary terms, that is simply a fantastic event. That's much better than winning the lottery, because suddenly you can reutilize glucose, you can start to grow, and you will not be lost. And just to have such a repair function is really worth to sacrifice 90% of your population to bacteriophages. And why do I say global scale? We have a world with 10 to the 31 bacteriophages. 1 in 100 of them is a transducing particle, so 10 to the 29. And the transfer is immense. So even if the frequency for an individual gene is so low, when you have 10 to the 29 particles, this suddenly becomes very, very relevant. So why I'm harping on about that? What I really want to say is transduction is not the same as lysogenic conversion. That's simply two very different mechanisms. One is really acting on a global scale, repairing bacterial genomes. The other can be potentially dangerous because it can spread antibiotic resistance gene or toxin genes. Unfortunately, there has been a mix-up. Some scientists talking to FDA mixed the two terms and said, our phage is safe because it doesn't do transduction. And now this has snowballed. We find this in documents not only by the FDA, but also by EMA and by EFSA. Everybody says, please find a bacteriophage that does not do transduction. And that's simply not possible because every phage does do transduction. Some do it with a little bit a higher frequency, others do it with a little bit a lower frequency. But as every bacteriophage has a lytic growth cycle and every bacteriophage packages DNA into head, they all do it. And we cannot find one that doesn't. So I don't know if it's possible to correct this mix-up and switch from transduction to lysogenic conversion, but let's see. And that's what I wanted to talk about today. As a conclusion, I reformulate or repeat my title. Is every bacteriophage suitable for phage therapy? We now know no, but we know what to look for. And we also have a much better understanding what could have gone wrong with earlier experiments. If perchance they use the temperate phage, it's clear there will be no effect. We also not only know which phages to use, but we also know how to grow them to generate safe preparations. And we think phage therapy should come back and that science is no longer the limiting factor. And with that, I thank you for your attention, and I'm very happy to answer questions.
Thank you, Hans-Jörg. And you did it on time, so congratulations. And such a complex topic explained in a very, very structured way. Yeah, a lot of congratulations coming, which is great. You can now take a sip of water, and in the meantime, because we have indeed a number of questions, so I will share them with you so that everybody can see them. Just a moment, because there is another one that just popped up. Okay. Okay, so I'm sharing with you the questions. Can you see them? I think so.
Yes.
So the first question, Hans-Jörg, I will try to read them, and you will try to reply. So the first question is, what is the difference between the genomes of lytic and lysogenic or temperate phage?
Okay. The lytic phages in principle are the simple ones. They only have this one mode of replicating. So the moment they enter a cell, this program to amplify and make progenies will simply run. The temperate phages are much more complex. They have to have the ability to stop this lytic program and choose another way. So size-wise, we do not find a big difference. So their lytic phages are very large, or temperate phages are very large. But the complexity of the temperate phage genome is simply much higher than the one of the lytic one.
Okay.
The question 2, no, there's no difference in shape. So you can have a jumbo phage that is lytic, you can have a jumbo phage that is temperate. And I showed you the morphologies, cifovirus or podovirus or myovirus, there's no difference. So from the shape, we cannot distinguish them.
Okay, shall I read number 3? When no strictly lytic phage is available against a target bacterium, to what extent could a temperate phage still be considered to reduce bacterial load in the first place, while waiting, for example, for trained phages?
I simply would not recommend to do that because you're basically shooting yourself in the foot. By using a temperate phage, you generate a completely new situation in the patient where suddenly this bacterium that causes the infection is also resistant against the temperate phage you're using. And this short-term reduction of 80% this is usually not enough to have a therapeutic effect. So I really, really strongly advise not to do that.
Okay, question number 4. If an antibiotic-resistant bacteria becomes lysogen bacteria, can it be susceptible to antibiotic, or it becomes a superbacteria?
The question is not really
clear.
So bacteriophages do not turn bacteria into superbacteria. So that's usually not the case. But of course, they can spread a toxin, or they can provide the bacterium with an antibiotic resistance marker, and that changes the bacterium. That will have suddenly a new function that it didn't have before.
Okay, question number 5. After treatment with temperate phages, do we know what proportion of surviving bacteria are lysogens, protected by repression systems, versus bacteria resistant through other mechanisms like receptor modification?
Okay, this idea that 80% grow lytically and 20% grow temperate or like a lysogen, this is an approximation that can vary very much between phages and also between conditions. But in principle, a certain proportion is turned into a lysogen, and then they will survive. Switching mechanism of receptor mechanisms, I didn't go into that. Some temperate phages actually, when they are integrated, they try to remove their own receptor from the surface. But that's not a phenomenon I managed to talk today about. So how can I answer the question? The proportion really depends on the genetic switch, how it is regulated, and how it reacts under the specific conditions. And that can, of course, be a bit different in the laboratory than it can be in a patient, but the effect is still the same.
Good. Question number 6. Do temperate phages maintain repeatability while plating them? Do they produce plaques all the time or stop after a couple of times?
Well, if you plate a temperate phage on its own host, it will never make a plaque. You will never see it. But if you plate the temperate phage on a host that doesn't have it, then it will always make a plaque like a lytic phage. So yes, this is reproducible. This doesn't suddenly stop, because that's the basic feature of the bacteriophage. It will amplify on a host that is susceptible to it.
Okay. We still have 3 questions here. We identified genes associated with a temperate phage lifestyle. Are there corresponding genes that encode lytic activity and classify a phage as strictly lytic that we should look for?
No, you cannot really do that. Because, as I said, a lytic phage has this entire program to amplify its genome and make new phage particles. But any temperate phage has the same program as well. So if you just find a gene for packaging, both groups have that. So you can really only exclude or say this is a strictly lytic phage by showing that it doesn't have any integration protein, that it doesn't have any repressor proteins, that it is not organized like all the lambdas or P22s. So that's just exclusion. Because, as I said, the temperate phage has all the functions to grow lytically as well. that's why the nomenclature is not really very nice. It's historically known and we're still using it, but it's a bit confusing.
Okay, next question. Regarding transduction by temperate phages, are bacterial genes that can be transduced mainly the genes flanking the prophage integration site, or can easily any bacterial gene be transferred?
Okay, I didn't go into details of the transduction. What I described is called generalized transduction that happens with a few, let's say with lytic bacteriophages, also with temperate bacteriophages, not a big difference. But there are a few forms of transduction that are very specific for temperate phages. And these are associated with problems of integrating the prophage, or rather with excising the prophage. So if this excision is not done properly, then suddenly the phage will have a gene, a bacterial gene that was flanking its prophage. That's called specialized transduction. Or you can have a problem that you actually start to amplify the phage genome, but it's still in the bacterial genome. And then you start to package this, and then you will suddenly start to package all the bacterial genome that is still linked to the phage. And that is called lateral transduction. But these are two phenomena that are exclusively associated with temperate phages. And as we are going to do phage therapy with lytic phages, I didn't address them in my talk today.
And the last question here, why is a single dose of phage not sufficient to eradicate the bacterial population?
I'm not sure if the answer is idea of self-dosing. Like the phage particles that come out of the infected cells should then take over the infection. This is usually not working very well, because in the natural environment, the phage doesn't amplify as efficiently as, for example, in the laboratory. And so the number of phage particles that is produced in that way is usually not sufficient to kind of simply dose itself. And that one dose is simply because you never reach 100% in a single go. Now, you might have an infection where you say, okay, if I reduce from 10 to the 8 to 10 to the 6, that already has a therapeutic effect. My immune system will suddenly manage and is stimulated by the cell debris and so on, and then it's going to work. But experience shows that several doses work much better than a single dose. And that's just the reason why we would use several doses.
And we have two additional questions in the meantime. So question number 10. So is bioinformatics the only way to find out if transduction happened or not?
No, you can do a nice transduction experiment. You just have to know what you're looking for. Like if you look for an antibiotic resistance gene to be transduced, then your donor bacterium has to have one. But you can do this experiment selecting for a glucose degradation protein, or you can do it for a single standard binding protein. Any protein, gene, let's say, any gene you can select for, you can make a selective plate, you can look if transduction happens. And that's the misunderstanding. Transduction is not a process to spread antibiotic resistance genes or toxin genes. It indiscriminately distributes all the bacterial genes among closely related species. And it's something that cannot be stopped. This happens all the time on a global scale. But it's really not dangerous. Just imagine you have this infection, I said. You have 10 to the 8 pathogenic bacteria, and if you want to do phage therapy, these are antibiotic resistant. And now you bring in a phage that has been grown on an antibiotic-resistant strain. So 10 to the minus 6 phage particles will carry an antibiotic-resistant gene. And that comes then in and repairs one of these 10 to the 9 antibiotic-resistant genes which are already in the system. What exactly does that change in the infection? It simply has no effect. For evolution, that's something else. For evolution, transduction is really important. But for phage therapy, it simply doesn't matter.
And the last question, somebody didn't get the connection of bacterial growth stage, log or others, and the change from lytic to lysogenic probability.
I don't know if how this understand how the genetic switch works is not clear. So let's just make a very simplified example. The phage wants to grow lysogen the host. So it has to start to produce a repressor. So the repressor molecules accumulate. And if enough repressor is present, it will switch off all the lytic functions. Now, if the cell is in a very bad state, like it's starving, then the protease activity in this cell is increased. And the proteases will start to degrade any protein, but among others, also the repressor protein. And so if the degradation rate by these proteases is higher than the synthesis rate, then all the repressor will disappear from this cell. And then the lytic functions can no longer be repressed. The phage will start to amplify its genome, late gene expression will start, and you go into the other direction. So this is what is happening in the cell. Now, the system is more complex than I describe it right now. You don't only have the repressor, you sometimes have an anti-repressor, and you have proteins that help stabilize the repressor, and you have functions that stabilize the other one. So this can go back and forth until a decision is made. I wrote a few literature to this genetic switch. If you're interested in that, it's well worth to read them. It's really very nice molecular system that decide between one or the other.
Perfect. Thank you, Hans-Jörg, for your presentation, for all the replies to a lot of questions. I see a comment here. I hope the recording of this webinar will be made available. Yes, it will be published on PhageEU website, and all of you who have registered to this webinar, you will receive an email with a link where to find these recordings. And on our website, you can also find recording from the previous session from October. So if you haven't been able to follow it, you can still watch it.